Good afternoon, everybody. So, to shake you from your postprandial torpor, we are engaged in an invigorating session on invasives, and it's good to see that we get quite a bit of coverage on the impact of invasives, which is often a neglected element of conservation science. So, our first speaker is Jan, who's going to be focusing on the impact of invasives on riparian plant communities.
Okay, welcome everybody. It's a pleasure to be here, and I'm glad that many people returned from lunch. So my name is Jan Čuda. I am from the Institute of Botany, Czech Academy of Sciences, and I will talk about impacts of invasive species on composition of native plant species here in African savanna in riparian habitats. My talk is a part of a broader or bigger project, and we studied different components of ecosystem as well as invasion, and I will focus here on botany, on plant species composition. We here are in Kruger National Park, and these protected areas act as barriers against invasion, and invasive species, or non-native species if you want to, can get into the park by many ways. One of them is along the rivers. That also can work as a habitat for them, because it's easy for invaders, for non-native species to establish in riparian habitats. We studied 3 invasive plants in this project, and all of them are from America. All of them are annuals. Xanthium strumarium was first recorded approximately 70 years ago, Parthenium hysterophorus 20, and Datura inoxia approximately 70 years ago. What's important that all of these invaders have not really studied impact on the ecosystem, and that's why their management is controversial. It's not clear whether they should be managed or not. How we did it? We established pairs of invaded and uninvaded sites within populations of these invaders, and it was 12 to 13 per each of them, and there were 100 square meters in size, and basically we determined all plant species there and compared the community composition by the means of ordination analysis. What we found was that there was, we estimated the effect of invaders on 2 components. The first one was abundances of plants. It means cover of other plant species, and the second one was presence-absence data. And we found that when considering just covers, the impact of all invaders when taken together was significant, and if we focus just on individual species, also effect of each of them was also significant. It was different for presence-absence data, where we found a significant effect of all invaders together. Then it was also significant for Datura and Xanthium, but not for Parthenium. And we think that it's because the Parthenium occurred on many different like habitat types, or more variable habitat types, and that's why the species composition was more variable, and that's why probably we haven't found any general trend for this. When we look closer, what happened? Here are 3 ordination plots for 3 species, Datura, Parthenium, and Xanthium, and there were more native plants were more common in controls, while in invaded ones there were more, in case of Parthenium and Xanthium, there were more invasive species, other ones, such as Alternatera pungens or others. On the other hand, some of the native species were more common, it means frequent or abundant in invaded sites. And it was probably because that this habitat of invaded sites was structurally different, and these species could be protected from grazing and so on. That's just the overview of the species that were more common in invaded or uninvaded sites. And so to conclude, most native species show negative response to the invasive dominant, but some of them were more frequent or abundant, as I told, in invaded vegetation, probably because of different structure and maybe also because of different microsite conditions, which will tell you my colleague Ananova after. And lastly, Xanthium and Datura mostly occurred in riverbeds, while Parthenium is spreading quite massively out of the riverbeds, so this is the species of consideration that should be managed. I would like to thank all colleagues and institutions that enabled this research, and if you would be interested, this is already published in Journal of Tropical Ecology.
Time for questions. So our next speaker is Damien, a 15-minute talk on an invasive wasp, which at this stage is impacting the Western Cape, but we'll see what happens next.
Good afternoon, everyone. So I'm Damien Gergon. I work at the Stellenbosch University in the biomath mathematical department and also in the genetic department. So today I'm going to talk about the yellow jacket invasive wasp Vespula germanica. So also this species is not yet in the savannas. Its highly invasive nature makes it a species to watch out because it's probably able to invade a large range of kind of environments. So Vespula germanica comes from the Palearctic region, mostly in Europe, but has already invaded a lot of different countries, USA, Canada, Argentina and Chile, also Australia, New Zealand, and recently South Africa. So the species is really invasive because of its diet. The species is able to feed on a large variety of food from, for example, small invertebrate and small vertebrate, sugar, a carbohydrate from fruits, and also human food and waste. So it allows the species to be able to survive in a lot of different environments. In South Africa, the species has been recorded for the first time 50 years ago in the region of Kirstenbosch. And the species has now spread across the Western Cape region, as we can see with the sample carried out by my colleagues. But however, what is really interesting is that the invasion seems low and limited compared to all other invaded regions. For example, in New Zealand, since the first introduction of the species, in 6 years, the species has invaded 80,000 kilometers square and is present in both islands of New Zealand. And in South Africa, in 50 years, the species has invaded only 7,000 kilometers squared. So we can ask the question, why is the invasion of Vespula germanica in South Africa limited and slow? If we manage to understand the factor that can limit the invasion of the species, it can be helpful to maybe prevent the species to come into the Kruger National Park, for example. So at the first step, the colleagues from my team wondered whether the species is theoretically able to invade other parts of the country. For this aim, they use distribution data from other invaded countries like Argentina and Chile, seasonal phenology data and irrigation data like rainfall, and they made modelization of the population dynamic of the species for South Africa. This is what they found. So they look for the suitable and non-suitable place for the species in South Africa. So as we can see, we are here, we are in a suitable area. So please, during the game view, after seeing a lion, try to find a wasp. I mean, if you find a wasp, it's coming to be a problem, but keep your eye open. But what we can see is that close to the recorded specimen, there is in red a lot of different, very optimal area for the species. But the species is still not here in this area. So there is a persistence of an invaded area despite an extreme suitability. So then, to have more information about the genetic diversity, Barbara Vanach and other colleagues from the genetic department of Stellenbosch made a study about the genetic diversity, about the wasp in South Africa. They use mitochondrial genome of sequence from sample from all the invaded area. And they build a haplotype network. So the principle of one haplotype network is to visualize identical sequence and to understand the relation between the sequence. So the identical sequence are called haplotype. So here we can see that there is two haplotypes in South Africa. So here the size of the circle corresponds to the number of samples carrying the haplotype, and the length of the branch corresponds to the relationships or the difference between the different haplotypes. So here there is two haplotypes in South Africa. The red haplotype, which is widely spread across the Western Cape region, but in low density close to the Table Mountain and Kinsterbosch region. And then there is the blue haplotype, which is restrained here, but very dominant in that place. So it suggests that they probably has two different introductions. The first introduction probably occurred in 1970s, but remains restrained in that region, where when the second introduction arrived probably in 2000, given the sudden expansion of the species in this region at that moment, and is maybe more adapted to the South Africa and is more spread. So the next question is, what is the reason for the difference in distribution between the two haplotypes? So in a very recent study, non-published yet, my colleagues from this time from the Conservation Department of Stellenbosch tried to determine the environmental differences between the zone of the two haplotypes. They hypothesized that there is a difference in adaptation to the new invaded area depending of each haplotype. So they use environmental variables like different measurement of temperature, rainfall, humidity, and they try to find some difference in the place of the different haplotypes. One of the results is that they found a difference in the temperature where the haplotype 1, where the haplotype red occurred and the haplotype blue occurred. So the haplotype 1 seems to live in a hotter region than the haplotype 2. So it is quite possible that the haplotype 1 comes from a gene pool more adapted to the hot temperature of the summer in South Africa. So the hypothesis is that, for example, the red haplotype may come from south of Europe or Mediterranean region where the temperatures are hot. and maybe the blue haplotype come from more the north of Europe when the temperatures are lower. So there is still a lot of unanswered questions that I want to try to answer. So the first time is there gene flow between the two haplotypes? What is the relation between the two haplotypes? So I want to investigate this mostly in the Stellenbosch region. So in this city, there is the particularity of having two haplotypes present. So it's a good opportunity to understand, for example, if wasps from different haplotypes can mate. And also the city is quite interesting because there is different road crossing the city and going to different part of the country. It's quite possible that the city is a starting point of subsequent invasion to other invasion in other part of the country because, for example, of transport. A queen can come into a car of a student, and when the student going back to home can bring the new queen in the new area. So the second question I want to answer is, are there any physical barrier to gene flow? For example, the mountain, the city? How can we learn more about the environmental condition in which the two haplotypes were identified? Can we have more precision about the environmental condition for each haplotype? And so formerly mitochondrial DNA was used, but mitochondrial DNA have the particularity to be transmitted only by female, so we miss a part of the genetic information. So it could be very interesting to use nuclear genome transmitted by both parents and have more precision about and better understanding the impact of male dispersal on the population structure. Finally, the last question is to understand all the routes of dispersal of the species. So what is the exact native area of the two haplotypes that we can find in South Africa? So to answer to this question, mitochondrial DNA was very useful, but as I said, it has a limited resolution. We cannot know the relation between the haplotype or infer the geographical barriers to gene flow. It doesn't allow to capture recent change, and we are studying recent change because the invasion is only 50 years ago, and we lack of information on male dispersal. So I'm going to use nuclear DNA. With nuclear DNA and some method, like WGS or RADSEC, we can have a higher resolution, capture the recent change, and have the contribution of both maternal and paternal. So I'm gonna use restriction site associated DNA sequencing for my project. Briefly, the principle of that method is to fragment the whole genome into small parts to obtain a large-scale genetic data, and I will be able to make the comparison between the two populations, for example, the population haplotype red and the population haplotype blue, and understand more the population dynamics of the species. So it can bring a more comprehensive approach to understand the invasion dynamic of the species and developing effective management strategy, maybe to prevent the species to come in a new environment like savannas. So my project also take part of a more wide project. The aim is to understand the route of dispersal of the species in the worldwide. So if some of you are interested in invasive wasp, my team is going to prepare a symposium probably in September, October in Stellenbosch in South Africa. So let me know if you are interested to attend and maybe to bring some sample from other country. And yeah, so thank you for the staff and for the organization, and Thank you for your attention.
Thank you very much, Damien. I think we've got time for one quick question. Okay, Corley.
Thank you for your talk. You might have mentioned this and I might have missed it in your introduction, but I was just wondering in terms of niche, how many indigenous wasps you have in Cape Town or in the Western Cape as opposed to Savannas, because you know there's quite a lot here. So I was just wondering if there's more space maybe for a wasp in terms of niche to come into an ecosystem like that.
So yeah, in the Stellenbosch region and in the Western Cape, there is hundreds of nests everywhere. It was your question. In the Savanna, at the moment, there is none. So we have some colleagues who look for, but at the moment, there is none, probably because the temperature is too hot. So it's a different niche, even if the modelization showed that this area can be invaded. But I think that if the propagal pressure is high, meaning if there is a lot of new crane arriving in that area, for example here in the Kruger National Park, there is a chance that a few of these queens find a place not too hot, in the shade, and with a good temperature to make a new nest and start a new invasion. So yeah, did I answer to your question? Quite off.
Great, thank you very much. Next up we have Martin, who's going to be again continuing the theme with invasives in riparian systems.
So, good afternoon, ladies and gentlemen. My name is Martin Heida, and as you may know, I'm member of the Mosaic research team, and the focus of my presentation will be on the impacts of invasive plants. So it will be botany. But if you feel sleepy after the opulent dinner or lunch, just go ahead. Of course, I did not do this work alone, but with a massive and substantial help from my friends. Okay, here they are. And as we all probably know, it's some already can be considered a bit of cliche, but it still needs to be repeated. The impacts of invasive plants are considered a major part of the human-induced global change and generally represent a threat to global diversity. It is interesting that this kind of statement is widely shared, but the impacts, especially in protected areas, are still not systematically documented. Further, riparian habitats represent a hotspot of invasion from the habitat perspective at the global scale, maybe because of the rapid propagal pressure or the massive disturbances induced by the flooding river, or by the constantly high or irregularly fluctuating trophic levels. Sorry. For this reason, we focused on 3 invasive aliens widespread in the riparian habitats of the Kruger National Park, which is Datura inoxia, Parthenium hysterophorus, and Xanthium strumarium. We adopted a space for time approach and sampled a comparative field data. This basically means that we were comparing the massively invaded vegetation with the adjacent uninvaded vegetation with presumably same or similar site conditions. This can be quite tricky due to the possible presence of confounded factors. However, the situation was a little bit easier for us because in the riparian habitats of the Kruger National Park, it's quite easy to find large homogeneous areas that differ in the level of invasion. We used general mixed effect models that to test the differences between the invaded and uninvaded vegetation. We used mixed models because to account for the spatial hierarchy of our sampling design. And we also used a mixed effect model ANCOVAS to test the differences in the response of native species richness to the aliens cover. We used native species richness, Shannon diversity, and pillow evenness as the importance values for our analysis. We detected significantly lower native species richness on invaded compared to the uninvaded plots. The difference was largest for Parthenium hysterophorus, and the differences in species richness between the invaded and uninvaded vegetation were non-significant for Datura and Xanthium. However, we also detected lower Shannon diversity on the invaded compared to the uninvaded plots. In this case, the largest difference, significant difference, was detected for Datura, followed by Parthenium, and the differences in Shannon diversity were non-significant for Xanthium. Similarly to Shannon diversity, we detected significant differences in PLO evenness, with the largest difference, significant difference for Datura inoxia, followed by Parthenium, while the differences in PLO evenness were non-significant for Xanthium. Here on this graph you can see on the left, too bad. No, okay, laser, that's what. On the left part of the graph you can see the response of native species richness to the aliens cover, while on the right you can see the response of alien species richness to the cover of our target species, because of course there were other aliens present in the plots or in the vegetation that we sampled. We detected marginally significant difference in the response of native versus alien species, as you can see by the black line. And when testing the effects of individual invaders, we detected a highly significant difference for Parthenium, which is the blue line, and the difference in the response of native versus alien species richness to the dominance cover was non-significant for the remaining two invaders, which is Datura and Xanthium. What to conclude? Out of the three invaders that we focused on, only Parthenium hysterophorus was detected to have a massive negative impact on native species richness. However, both Datura inoxia and Parthenium hysterophorus were detected to have a significant negative impact on Shannon diversity. However, in case of Datura inoxia, the impact on Shannon diversity apparently occurs without a significant loss of native species. Interestingly, the response or the differences between the invaded and uninvaded vegetation were extremely variable in the case of Xanthium strumarium, and our field experience suggests that in some cases its effect may be even facilitative, that it somehow stabilizes the riverbeds, and you actually find more native species in the stands heavily, or in the dominant stands of Xanthium compared to its surroundings. For this reason, we think that the Parthenium hysterophorus should be prioritized in management efforts, which is already happening, and should be carefully monitored, especially when escaping outside the riparian habitats. We conclude that the threat of Datura inoxia and Xanthium strumarium is relatively minor compared to Parthenium. However, these species these invaders should be carefully monitored, and eradication efforts may be needed, especially when they escape outside the riparian vegetation. And what to take home is that the impacts of invasive plants cannot be estimated from a distance. You always need somebody to crawl through the invaded vegetation and record the present native species. No matter what kind of scale you are working at, estimating impacts without the detailed knowledge of the invaded vegetation can be really misleading. This is mainly because there are apparently invaders that reach a massive dominance in the invaded vegetation. However, at the same time, they do not really suppress the richness of native species. I'd like to finish with the acknowledgements of our funding agencies and with the logos of our home organizations, and most importantly with the photo of our botanical field research team. Many thanks for your attention.
Many thanks, Martin. It is quite alarming to see just how much Parthenium is spread through the matrix in the south as well. Any questions? We've got a couple of minutes.
How do you control Parthenium?
I have very little experience with practical management of Parthenium, but as far as I know, it is controlled chemically by spraying. But I'm sure there are more competent people in this room to answer your question.
I think there's also a biocontrol agent, but it's tediously expensive and difficult to manage. Quick question, if there are none others from the floor. We've done quite a riparian forest regeneration work in the far north, and our impression, we didn't actually work on it, but our impression is that these invasives can also suppress the regeneration of tree species. Mm-hmm. Did you find any evidence of that at all?
This is difficult. We actually didn't focus on adult trees, but it's very likely that the tree juveniles cannot thrive in the stands of Parthenium, the same as herbal species. So from the long-term perspective, I would suppose so, that because it's not possible for the juveniles to grow there in the dense stands of Parthenium.
Magic. So if no further questions, then we've made up a little bit of time. Thank you very much. So we've heard a lot about biocontrol agents in the past, but now we're switching to some novel work on looking at how, so Jaco is going to talk to us about how native herbivores might control invasives.
Good afternoon. I think many of you might be disappointed to learn that the herbivores I'm talking about would not be elephant, hippos, or rhinos, but rather these very good-looking little guys over here, soapberry bugs in the genus Leptocorus. I'd like to start by acknowledging my co-author, Scott Carroll, is from UC Davis in California, and Levi Brown and Dylan Garrity are two master's students who did most of this work that I'll be talking about today. I'm going to take you to the far north of Australia, the tropical savanna areas there, and hopefully convince you that we can measure evolutionary responses in native organisms to invasion. So a few of us have argued for a long time that we need to think about interaction ecology really when we want to think about rapid evolutionary changes in novel interactions such as those between native and invasive species. So the eco-evolutionary experience is simply a term that describes the adaptations, or in the case of introduced organisms, pre-adaptations, due to its interaction ecology over evolutionary timescales. But this can have a very significant impact on evolutionary responses over ecological timescales. So I'm talking about hundreds or 200 generations. I'll give you one example. If the invasive species has very high eco-evolutionary experience with the native species that lacks eco-evolutionary experience, you may expect selection to be quite strong. For example, in Australia, cane toads are infamous as one of the most devastating invasive vertebrates. They're also toxic to things like snakes. And lots of Australian snakes lack triple E to toads historically. Cane toads, the level of poison that they produce is also scaled allometrically. So the largest, fattest cane toads produce the most toxin. And subsequently, some snakes have evolved smaller gape size because they get knocked out of the ones that are capable of eating those cane toads get knocked out of that gene pool. I'll explain why high triple E of both the invasive species and a native species matters for high selection strength using my own system that I'll be talking about. This is Leptocorus gallicus. It's a mipteran native to Australia and some of the areas around Oceania. And here it's feeding on Alectryon tomentosus, a plant in the family Sapondaceae. These insects are polyphagous, but they are specialist feeders on a certain tribe of plants in the family Sapondaceae. So they do share lots of eco-evolutionary experience with sapondids. The bottom picture here shows one of the introduced invasive plants that I'm working on, Cardiospermum harlicocabum, and it's also a sapondid, and therefore has been colonized by this insect in Australia. Now you can imagine that invasive species by definition would be highly abundant, widespread in most instances, and therefore they can become either uncontested resources or a threat in the case of something like predators. So just a bit of background. In Australia, we've got two of these balloon vines in the genus Cardiospermum that have been introduced. The one is harlicocabum and the other one is grandiflorum. And there are interesting differences in the invasion histories in the country. You can see harlicocabum has been there for a very long time and indeed has been noted by the earliest florists of Queensland and the Northern Territory. And grandiflorum has been more recently introduced only about 100 years ago and is mainly restricted along the east coast of that country. In both instances, these invaders have been colonized by Leptocorus gallicus. These insects obviously still utilize some of their native host plants, and I'm just illustrating two of those main host plants that we are aware of on the interior and on the coast. But what I want to illustrate is that you can see that the challenges that these insects on the new host plant face are quite significant in terms of the spatial configuration of seeds. So in other words, getting to the seed of a balloon vine may require you to have a much longer beak or proboscis than getting to the seed of a native woolly rabbitane or Alectryon. So we sampled insects up in the north end, close to Darwin and south of Darwin, and along the east coast of Australia to do some morphometric analyses and genetic analyses of these insects. Our data confirm earlier reports by my friend Scott Carroll, which showed when you look at sympatric populations of host plants in the environment, and we do these comparisons between the native host plant and the invasive host plant, when you compare the fruit radius, in other words, the size of fruit, to the beak length, you can see in this instance almost a 38% increase in beak length. And by the way, we had these insects for multiple generations in the glasshouse, so we know that this is genetic and not just environmental. For the comparison on the east coast, when we compare the more recently introduced grandiflorum to its sympatric populations of the main native host plant, we see an increase which is statistically significant but much less pronounced than in the case of this old established invasive species, only about a 10% increase in beak length. So we genotyped all of these populations at about 10,000 SNP loci to get an idea of population genetic structure. And the main take-home message here, which I would like you to focus on, is that the insects from the Northern Territory all the way up there in Darwin is genetically quite differentiated from everybody else on the east coast, and those populations on the east coast, very low genetic differentiation. So on different host plants, in one instance high genetic differentiation and low in the other. So with this in mind, we wanted to first explore whether insects actually have a higher fitness on the new host plants. So why are they seemingly evolving longer proboscides on their new host plants? And then we wanted to do some breeding experiments. So we wanted to test basically whether these two different levels of genetic differentiation, which is likely to do with the residence time of these new host plants translate into some fitness trade-off. So incipient reproductive isolation, if you will, in the case of these highly differentiated insects. So for the diet experiment, we kept these insects for a few generations in the lab on the host plants that we collected them, so the diet, and then we split them in a second generation either onto the same diet or either the native or the invasive host plant. And the short answer is diet matters. So it didn't matter whether we switched these insects around, they always had a much quicker development time when they were fed the invasive diet, so Cardiospermum grandiflorum. We also worked out a correlate for relative fitness, which is simply the number of hatchlings that each individual pair had compared to the fittest genotype or pair of genotypes. And again, what you can see here is this relationship holds up. So the relative fitness of insects fed the invasive diet is almost 3 times that of insects that were fed their native diet. For the second experiment, this is again now just trying to look at differences between those genetically differentiated versus not really differentiated populations. We grew them for a few bred them for a few generations in the glasshouse, and then we had a parental population of a control line, so Helicocabum insects, grandiflorum insects. Again, these are the 2 invasive hosts, and then one of those native host plants, tomentosis, Electrion tomentosis. We created filial 1 crosses between these breeding lines and filial 2, and then we measured again fitness. Now this is a lot of work. For somebody who normally works on plants, I got to learn very quickly raising insects and counting eggs is no easy task. And therefore, this is quite a disappointing outcome. After all of this work for these genetic lineages that seemingly are very differentiated, we didn't find any difference in fitness between the hybrids and the parental populations on those different host plants. The same is true for the, obviously, for the populations that showed low genetic differentiation, but these included the insects where we had a diet treatment included. But again, it illustrates that the invasive diet is really good for these insects. They love it. So when you breed so many insects and you measure them, so you measure the parents, you measure the offspring, it gives you the opportunity to kind of get more to a quantitative estimate for heritability. In this instance, there's a very simplistic way where we just regress the values of the offspring for beak length, for example, against the mid-parent value. And the slope of that regression gives you a very rough estimate of heritability, which turned out in this instance to be about 60%. Now, if there's any quantitative geneticists among you here, you would say, well, this is actually not a good way of estimating heritability. So we subsequently ran these data through an animal model where we take into account the pedigree and all of those traits, and heritability remains really high for beak length. In fact, so high that when we do an artificial selection experiment in the lab, which we've been doing now for only 6 generations, we could increase beak length by a further 10%. So we now have insects in the lab that has 20% longer beaks on average than the insects found in the field on the native host plant, in the case of the grandiflorum insects. And there are lots of reasons why we want to do this, and I can't go into the details, but you are welcome to ask me afterwards. So, despite high levels of gene flow, we see strong directional selection on beak length due to the utilization of invasive host plants. And we are arguing this is partly due to the nutritional benefits that we see in the lab. So in other words, these insects seem to lay more eggs, have more offspring, and those offspring develop quicker. Our ongoing work at the moment is to assess differences in plant host toxicity. There's lots been written about cyanide compounds, for example, in sapondaceous species. So the next step is to look at differences in toxicity of the seeds of those different host plants. I also have a master's student currently working her way through about 300 hours of video footage where we provided these insects with choices between different host plants to see what choices they make and how long they feed on different host plants. And then, of course, we are really interested to see how far we can push this evolution of beak length in the lab, because at some point you may expect trade-offs to occur. When beak lengths get too long, other things may be getting too wide or too short or something else. So I'm really excited to be in Kruger because I spent a few weeks here, or a week or so before I came to this meeting, just running around Skukuza mainly, collecting, again, this invasive host plant that I just spoke about in Australia, Cardiospermum hylicacabum, but also a native Cardiospermum species that occurs in the park, Cardiospermum corundum. And I've measured now in the last week about 200 or so insects, and I wish I had time to actually crunch the data, but I haven't had any time, so you'll have to wait until next time before I can tell you what's going on. But it's certainly pretty obvious that there's a massive difference in the sizes of the fruit of these individuals. And therefore we're thinking something similar might be going on. Why? Because Kruger is also home to about 4 different species in the genus Leptocorus. This is a native species to the area, Leptocorus mutilatus, which is the one that I focused on for this particular study. So I'm hoping to update you sometime in the future. We've also sequenced the full genome of Leptocorus gallicus, which is currently being annotated, and the main idea here is for us to look for convergent patterns of evolution, potentially between different continents, so between Australia and Africa, which I think would be a fascinating finding if it holds up. That leaves me only to thank my collaborators and all of the students and helpers who worked very tirelessly to gather all of this data. And if there is time, I see there's 2 minutes remaining, I can answer some questions. Thank you very much for your time.
So thank you very much. Fascinating and a huge amount of work. So, and really impressive artificial selection there. What do you think is maintaining the genetic variation in beak length in order to allow it to evolve so rapidly in response to selection?
That's a good question, and I don't think I've got a straightforward answer for you, but I can tell you what I'm thinking. So in addition to those plants that I discussed now, there are numerous other sapindaceous species that these insects naturally feed on in Australia, and they all differ in fruit size. And we think what is actually happening is at the beginning of the fruiting period, when these seeds are getting ready to be eaten, essentially, insects will fly in. And we think initially, because many of these sapindaceous plants, as you know, the capsules will eventually break open and the seeds will be available. But it would be really those females in particular that comes first with long beaks that will have the real advantage over those females with shorter beaks. And it might be not true for other host plants where you have very small fruit. So we think it's this kind of fluctuating selection that's going on in the field. Yeah.
Magic. Thank you very much, Yaku.
Thank you.
So moving on from sexy insects to something a little bit more familiar, Lisa's going to talk to us about other forms of alien control.
Thank you very much. I decided to wear this thing because I'm going to be looking away all the time. So I want to tell you a little bit about some of the work that several of my colleagues and I have been thinking about over the last couple of years, and it's really about the functionality of novel ecosystems. And when I talk about functionality, we're thinking really kind of broad functionality, so not functionality, sorry, novelty. Novelty, broad novelty in terms of species compositions and maybe structure or climates. But I'm going to focus today on I'm going to focus mainly on novelty in terms of species assemblages. That's both driven by the introduction of, like, human-driven introduction of novel species, but also about distribution shifts that results because of environmental change. So, and Jens Christian Svenning has recently received some funding for a center, a 6-year center, specifically to be asking these kinds of questions. So essentially what we want to know is, what are the processes that must be in place within an ecosystem that will ensure a continuation of functionality within that system despite novelty? And so I want to show you a little bit of the work that led up to the formation of this center, and then also some of our plans, particularly in South Africa with the startup of this center. And just to point out that most of what I'm going to show you was led by Ninad Mungi, one of the postdocs in our lab. All of the India work was his leadership. Okay, so one of the key considerations when thinking about novelty is that it is probably an inevitability of the future, right? The latest IPBES report that came out on invasive alien species showed that they estimated about 200 new established alien species recorded per year globally. And then the other big issue is that future climates are almost certainly going to be warmer than they have been for millions of years. So that graph in the corner there, that's the reconstructed temperature trends for the last 40 million years. And it's projected forward into 2300 with the 3 different climate scenarios. And you can see that we're entering a climate zone that the Earth hasn't experienced in a really, really long time. And the little insert pictures above that graph shows the difference between the vegetation biomes that used to be supported under previous temperature regimes compared to what it is today. And I hope you can see the little tracks there, because it also then links it to how close in the future we are to that temperature that supported such a vastly different vegetation biome. So the point being that it would be safe to assume that we will have very, very different species assemblages in the future. One of the other key messages that came from this IPBES report was, well, so one, that biotic homogenization is one of the major negative drivers of this negative impact on native species diversity and functionality of the system. And that's because oftentimes when things are arriving into a new system, especially the very aggressive invaders, they end up just proliferating, and they achieve this single species dominance that you can see in the corner right picture there. But the other really important thing that I think to take from that IPBES report is that despite the fact that they recorded more than 37,000 established alien species globally, there's only 3,500 that they list as having documented negative effects. So clearly there are a lot of, there's a lot of scenarios where these species do not achieve this single species dominance. So there are clearly conditions under which you can maintain or you can curb this tendency towards single species dominance. And we, of course, have our suspicions. And that's one of the things that we really want to pursue, is to look at how the trophic impact and the intactness of a trophic, of top-down trophic impact and disturbance can curb this single species dominance of novel plant species in the system. So that brings me to Ninad's study. He asked exactly that. He was wanting to know how mega herbivores in India could perhaps provide that resistance towards the single species dominance. And so he assessed the relationship between the abundance of mega herbivores and the native and alien plant richness and cover across much of India. And he used this amazing study or data set that the Indian government's National Tiger Conservation Authority and the Wildlife Institute of India collect to look at the country's tiger populations and their habitat suitability. And the survey that Ninad had used was collected in 2018 to 19, and it was such an impressive effort because they essentially had 141 sites across the whole of India, and they chose the sites to be within what they call like sites that have a high wilderness intactness index. And within each of these 141 sites, the sites were at least 200 square kilometers large, and then they put a grid of 2 by 2 kilometers onto each of these sites, and then a pair of cameras per grid cell. So they ended up having almost 27,000 camera stations, and they effectively sampled over 120,000 square kilometers. This study actually got into the Guinness World Book of Records for the biggest trap camera study ever. But then at the same time, they had vegetation surveys where they monitored native species richness, native species cover, and then also aliens. And this was on a 25 square kilometer grid where they put 1 to 8 2-kilometer transects. And And then they had this concentric circle design where they measured all the trees in the 30-meter circle, shrubs in the 10-meter, and all the grasses in the 1-meter. And this was on every 400 meters of this transect, so they ended up with almost 160,000 vegetation plots. So an absolute immense effort. And so this is the relationship between what Nina had found between mega-herbivore relative abundance and then the plant variables. So so you have rainfall on the right, and then mega-herbivores in the center, and that's an intactness index, because they also evaluated how many signs of human impact they could see at each of these sites. And so the green vegetation represents the native vegetation with richness at the top and cover at the bottom, and then the brownish pink is the invasive, or the alien, not necessarily invasive, alien plant species, again with richness at the top and with cover at the bottom. And so what they found is that with rainfall always included or increased plant richness and plant cover, regardless of novelty. So both novel species and native species, rainfall increased it, and mega herbivores increased native richness, and it increased native cover. And that positive relationship on native plant species from mega herbivores were most pronounced in the intermediate rainfall, intermediate productivity regions. But then also what we found is that the presence of mega herbivores had a negative impact on the alien plant cover, but it had no relationship with alien plant richness. So essentially, what this suggests is that when you have a high abundance of these mega herbivores in the system, they are reducing the cover and reducing this tendency towards the single species dominance. They might not be making any difference to which species remain in the system, but they might be disrupting this homogenization from some of these aggressive invaders. And then just a little caveat is that Nina had also found that if you categorize the alien species into categories, one of which would be the thicket-forming growth forms, then the thicket-forming in aliens, once they reach a level of 40% cover, then this ability of mega herbivores to disrupt the system started to decline. So, all right, so that was really one of the first studies that started leading to this application for the center to look at this question in a lot more broadly. But we are not the only people to have found similar results. There's been a couple of papers recently that show similar kind of mechanism of large mammals curbing this tendency for these plants to just take over an area. And so it's leading us to hypothesize that if we assume that the future will hold novelty, and novelty in terms of species assemblages, then without herbivore impact, without the disturbance that they provide, and then we would expect that there should be a shift in biodiversity, and we would probably lose some of the species from a system, and with it, you would expect a shrinkage in the functionality of the system. But what we're hypothesizing is that once you have animals within the system and you maintain this disturbance feature and this constant creation of extra niche space that then supports the native plants, then you might still have a shift in which species are there. It would still be a novel system, but you might maintain a lot more of the species and a lot more of the functionality, or a lot more of the diversity and a lot more of the functionality. So essentially, if this is where we were before in terms of if you have a biotic novelty and an abiotic novelty axis, and we have already started moving up to this, down this line of having novelty in the biotic and the abiotic components of the system. And eventually we'll get into this quadrant 2 where things are very much different from where we started. But if we keep restoring previous assemblages, then of course we're pushing the system back into this 4th quadrant where you have a mismatch between the environmental conditions and the biotic composition. All right, so that all led to this development of this center. It's called Econovo, the Center for Ecological Dynamics in a Novel Biosphere. And essentially what we are trying to do here with all these 4 different packages is that we want to, we're hypothesizing that the dynamics in the diversity dynamics within novel systems are not universally negative, and that we're trying to find what are those drivers that maintain that functionality, and we want to see whether the restoration of complexity within these systems, also complexity in terms of the trophic impact, but geodiversity, etc., would maintain this resilience and functioning of the system. And so we've already gotten started. We have 2 PhD students, Jonas Triepel and Londiwe Mkuna, and they are, Jonas has a project where he is now trying to look at this, he's trying to find areas that vary along this gradient of trophic complexity and trophic intactness, and then he'd be measuring the novelty in terms of plant species and the biodiversity that is maintained within that system. So he's at the point where he's trying to find areas that represent these gradients well, the gradients in novelty and the gradients in trophic complexity of large mammals. And then Londiwe focuses in more on the plant species itself, because of course not every species will lend itself to this kind of control by mega herbivore disturbance. So she's looking more at the traits that characterize a species as susceptible to this kind of control, or perhaps some species would just benefit from dispersal. And so, Londiwe is also co-supervised by Michelle Grieve. By Michelle Grieve, and she's based at University of Pretoria, and Jonas is based at Aarhus University. So we are actively looking for people wanting to get involved in these kinds of programs. If you have sites that you know of that fit well into these questions, please come and chat to me. I think I am out of time, so I will just say the take-home messages is that if we embrace novelty to a certain extent, we might have the capacity to steer these systems into functional ecosystems, and use our understanding of biodiversity dynamics to know what will make a novel system continue into the future. Thank you very much. I put an ostrich there because I also want people that work on ostriches to come and speak to me.
Thanks very much. I'm afraid no time for questions, but thanks a lot. Just to draw your attention to 2 posters outside. Claude and Kensani, please do have a look at those posters when you have a moment. Our next speaker is Anna, who will look at trophic cascade effects of invasives.
Sorry, I move a lot, so if I have to speak to the mic, nobody will listen or will hear anything. Okay, so hello everyone. Thanks a lot for staying here still. I'm Ana Novoa. I'm based in the Institute of Botany of the Czech Academy of Sciences, so I'm part of the same group of Martin and Honza that I already talked before. And I'm going to continue with the project that they already mentioned before, but this time I'm going to be talking about the impacts of invasive annual plants in soil characteristics and microbial communities. There you go. So, as my colleague said before, the invasion of alien plants are a big threat for the conservation of biodiversity in Kruger National Park. So here you can see a figure that was published by Llewellyn Foxcroft and colleagues last year, and it indicates the number of new records of alien taxa, of alien plants, in Kruger National Park over the years. And you can see that these records have been increasing. Nowadays, there are 146 alien plants present in Kruger National Park already, and already some of them have become naturalized and invasive. So it's a big threat. And among these 146 species, there are 3 that are particularly controversial, and I guess you cannot imagine which ones are they. We go back to the same, to these 3 annual plants. So Parthenium hysterophorus, Datura inoxia, and Shantium strumanium, they are all native from the Americas. As already Martin and Honza said before, they are annual plants, so this means that during the growing season, they are alive, and they occupy large areas, they dominate the invaded community, but after the growing season, they die, they decompose, they disappear, and therefore it's not clear, or is becoming now a bit more clear, whether they have important negative environmental impacts in Kruger National Park. And therefore it's also not clear whether they should be prioritized for management or not. And this is why we established this project, to try to answer these questions. Also, they invade riparian habitats, my colleagues already said before. And rivers play a really important role in the invasion of alien plants in arid areas. Because the level of water changed over the year, open spaces appear that they are susceptible for the invasion of alien plants. And in fact, our 3 target species, they tend to occupy all these areas in the growing season. However, the dynamics of these invasive annual plants in rivers in arid areas are not really well studied. So we established this project trying to understand this impact and invasion of annual plants in Kruger National Park. For that, as Martin and Honza already mentioned before, we established different sites invaded by Parthenium, Shantium, and Datura in Kruger National Park. So we have the sites invaded by Parthenium in the south, the sites of Shantium in central north, and then the sites invaded by Datura in the north. And in each of these sites, we established pair plots invaded and non-invaded by the target species. So here in this picture, you can see some examples. A will be a plot invaded by Datura. B will be the paired uninvaded plot. The same with C, invaded by Datura, uninvaded pair plot. E, invaded by Parthenium. F, uninvaded pair plot. And G, invaded by Shantium, and the H, the pair uninvaded plot. Now, with this setup, what we did was to follow different methods to study the different levels of the invasion, which will be these pieces of the mosaic. So we did aerial surveys, which Peter will be talking about them later, botanical surveys that Hans and Martin mentioned before, herbivore surveys together with DNA metabarcoding, which Clara will be talking about later, and soil surveys together with DNA metabarcoding that I will be discussing now. So getting into the soil, I'm sorry, it's DNA metabarcoding again, but this time it's bacteria and fungi, so it's a little bit different. So what we did here was to establish these 7 sites per species, so 7 sites invaded by Parthenium in the south, Shantium in the central north, and Datura in the north. In each of these 7 sites per species, we established these pairs of invaded and uninvaded plots, and in each of these plots, we assessed soil pH, humidity, nutrients, enzymatic activities, and bacterial and fungal diversity and composition. We did this in January and April because, as I said, they are annual plants. So in January is the peak of the growing season. They are really happy and occupying large spaces and really big. But then in April, they are already decomposing and dying, and we thought it would be interesting to compare these 2 seasons. So now moving on to the findings. I'm sorry, it's a lot of numbers. So I'm going to walk you through them. So this table shows the results for the soil characteristics and soil enzymatic activities for January, and is divided by the sites of Parthenium, Datura, and Shantium. Now, for soil characteristics, we look at, as I said, pH, humidity, phosphorus, carbon, ammonium, nitrate, and calcium, and then enzymatic activities, phosphatase, beta-glucosidase, and urease. Just to clarify, these 3 enzymatic activities are linked to the cycles of phosphorus for phosphatase, carbon for beta-glucosidase, and nitrogen for urease. Now, what we did was to compare invaded and uninvaded plots, these pair plots that we have. So in January, we found significant differences in phosphorus, calcium, and urease activities. Basically, we found higher levels of phosphorus and calcium in invaded areas for Parthenium and lower levels of urease activity. This might be telling us that although Parthenium seems to be adding phosphorus to the soil, those microorganisms that are linked to the cycle of phosphorus don't seem to be reacting to it. And on the other hand, the microorganisms responsible for the cycle of nitrogen seem to be not so happy about the presence of parthenium. On the other hand, we didn't find significant differences for datura and xanthium. Now moving to April, we only found this effect on calcium that persisted when the plants are already decaying and decomposing. Now moving to bacteria and fungi. As you know, bacteria and fungi are the main soil microorganisms that we can find worldwide. So we look at them. This is just a visual representation of the most common classes of bacteria. So here in the legend, although I'm not sure if you can read it, but it's not necessary, you can see that the phylum and the class of the most common bacteria, and here you can see the abundance, relative abundance of those bacteria. And of course, like these too many colors, we cannot see many things, but there are some things that are interesting, like for example, these light green bacteria over here, which only appear in January in datura and xanthium areas, which makes sense because these bacteria are actually linked to the presence of water. January is the wet season, and datura and xanthium sites are normally closer to the river than the sample parthenium sites. Also with fungi, what we can observe here easily is that this, for fungi, there was a little bit less variety in classes. But that was for abundance. Now, if we look at number of OTUs, probably you cannot see it so well either here, but we have the number of OTUs found in parthenium, datura, and xanthium sites here on the top. And what calls the attention at the first look is that we have lots of OTUs here. So these are the OTUs that only appear in uninvaded sites in January. So we have a large number of OTUs for parthenium in uninvaded sites. While for datura and xanthium sites, we have higher numbers in invaded sites. So it seems to work in the other way around. Now, exactly the same numbers, but we break them into the uninvaded areas, and we have the different colors for datura, parthenium, and xanthium sites. And we can also see a high number of OTUs that appear only in parthenium sites. But then when we go to the invaded areas, the OTUs that appear only in parthenium sites are much less. So obviously parthenium is doing something there. If we look at the same with fungi, we see exactly the same trend. So a high number of OTUs linked to uninvaded areas in parthenium, and a large number of OTUs linked to invaded areas with datura and xanthium. Now we break them down in uninvaded and invaded areas, and once again we see a high number of OTUs linked to parthenium in uninvaded areas, and the number shrinks when we look at the invaded areas. So something is going on there, but this is only a visual representation. Let's look at some analysis. So we calculated different indexes of diversity, and here you can see the results for January, the results for April, and once again, they are presented for parthenium, datura, and xanthium sites, comparing invaded and uninvaded areas. And we only found significant differences in April for the exponent of Shannon, which was higher in invaded areas. So it seems that there is not a big impact, it seems, in diversity of bacteria. And in diversity of fungi, we didn't find any significant differences. So we could think, okay, the impact might not be that big. However, when we look at the composition, there are clear differences. We found significant differences not only between species, which kind of makes sense. The parthenium sites, which are in the south, are really different to the xanthium and datura sites, which are in the north and not center, and that was regardless of status and regardless of season. But we also found significant differences between invaded and uninvaded areas. So there is an impact in the composition of bacteria, and exactly the same for fungi. We also found significant differences between status, regardless of the season or regardless of the region or the species. And just finally, we also did an analysis to find indicator species, so meaning species or OTUs that are specifically linked to particular areas. So this will be for bacteria, xanthium, parthenium, and datura, divided in invaded and uninvaded areas, and the same for fungi. And we found lots of indicator species for invaded areas, but also for uninvaded. And this starts to show us some interesting things. And just to mention some, we found in parthenium sites that these bacteria, Nostocaceae or Nostocaceae, which are known for their nitro-fixing abilities, they are an indicator species for uninvaded areas. Actually, if you look at the relative abundance, in uninvaded areas, they have 0.086% of abundance, and in the invaded areas, 0.003% of abundance. So they are more common in uninvaded areas. And if you remember, for when we look at the soils, the urease activity, so the activity of microorganisms related to the cycle of nitrogen, was also lower in invaded areas. So there is something going on here with Parthenium and the cycle of nitrogen. So just some take-home messages. Only Parthenium appears to cause an impact on soil characteristics, the activity of soil microbial communities, and the diversity of soil bacterial communities. However, all the target species, the 3 of them, impacted soil microbial community composition in both seasons, so regardless of being alive or being decomposing. So this, together with the results that my colleagues presented before and the others will present later, tell us that action is needed, and especially to control invasions by Parthenium in Kruger National Park. With that, thanks a lot. Of course, this was not only me, or the whole team, so thank you to all of them. Thank you to my family to always come with me to the field work, to do the field work, and thank you to all of you for listening.
Thank you very much, Anna. Any questions?
Yeah, thank you very much, Anna. I was wondering, are the stage of invasion of these Parthenium fields, do you know how long they've been there? Is there knowledge on, are they early stage invasion, or have they been around for 5 years and now you go in? That might be quite important for your composition.
They have been there for a long time. Yeah, Martin, I don't remember exactly now the year, but Martin showed before the year of introduction, and these invasions come over every year, yeah, so they have been there for a long time. There you go.
Any other questions for Anna?
Thanks so much, Anna. I was just wondering if you measured the right parameters in the soil. And the reason why I say this is, I don't know if you can kind of match up those bacteria with processes that happen in the soil, such as, nitrification or mineralization, and then, things that might encourage nodulation in species. And then also, you haven't looked at specific organic molecules that might, that might have allelopathic effects. So I think just because you haven't found it in the things that you measured is maybe, there's many more things that you can look at that might affect those processes in the soil. Oh yeah, for sure. So there are so many things. You mentioned allelopathy. That's interesting because Parthenium is believed to be allelopathic. There are some studies in Kruger from some years ago, but they concentrated, so they extracted concentrating Parthenium in water, and then they checked that it was allelopathic, and that's not the natural way that you should do it, so that's something that we should check in the future for sure, not only for the soil, but also for the plant community to see if it has allelopathic effects. Of course, there are so many more things that we could measure. Actually, of the soil characteristics, we measure many more. I just didn't present all of them. We had I think 30 different variables. I just presented the most common ones and the ones that are more linked, so carbon and nitrogen and phosphorus. But of course, there are so many more things that can be impacted by Parthenium or Datura and Xanthine for sure. Yeah. These are just the most common ones that we normally measure. It's the first step. We don't know anything, or we didn't know anything that was going on in the soil. Thank you. And about nodulation, you mean affecting nodulation to native species? Because these species do not... Yes, I was just thinking, if you have certain bacteria that is negatively affected by your invasive species, but that would affect nodulation or some kind of transformation of nitrogen in your native species, then that would have an effect. But that's not so clear in the data that you presented. Well, it's in the indicator species, so we now have to see whether the indicator species correlate with any of these characteristics. I must say I just analyzed this data last week, so it's still an ongoing process. Thank you.
Magic. Thank you very much, Anna. And our next speaker, a 5-minute talk, is Peter. Again, just thinking about mapping effects or distribution using aerial photos.
Good afternoon. As Anna mentioned, I will be another speaker talking about on the same project, and you will get even more from Clara afterwards. And my talk will be a little different from the others because first it's short, it's only 5 minutes. It's not really on impact. I will rather talk about the distribution, which is nevertheless a basis for, let's say, large-scale estimation of impact magnitudes. And also, I won't really show you any real results because we acquired quite a big amount of data that is still processed, but I hope to show you what we are up to. We stick in the same habitat, riverbeds in Kruger, but we will look from above, because my part, what I'm going to talk about, will concern the aerial survey, aerial monitoring. And this project is a follow-up of the Mosaic project that we did before here in Kruger, where we looked at the species richness and diversity of multiple taxonomic groups in habitats, in different habitats across Kruger. And we also touched the invasive species, but this project has already invasion focus. And the rationale here is that the species get into the park from the outside, from external surroundings of the park. They establish in the riverbeds and spread from riverbeds to the open savanna. And the focus here is on impact. And in this part, these are our 3 invaders. You've heard enough about them. And in this part of the project, we actually be using light aircraft to map the occurrence of these invasive species along the 5 main rivers, perennial rivers if you want, across the entire river flow from west to east, and we do it up to 500 meters from each riverbank to capture the spread from the riverbed to the surroundings. And we also included 20-kilometer sections before the river enters the park to account for what we call propagule pressure, and it means actually the particles of plants that can give rise to new population, and in this case, because they are annuals, it's seeds. A little bit technical information. We acquire orthophoto at 7 centimeter resolution in 10 multispectral bands, and we use models of vegetation height to ease the identification. That's done by machine learning techniques, and the aim is to get this area where the invasive populations occur. And the aim, what we hope we will get, is this picture where the invasive population is mapped, and it will be done across the whole river flow. The second aim of it is that we identified particular sites where these species occur, and we will do repeated sampling to get idea about the interannual dynamics of these species. So if we look at Parthenium, start with Parthenium, on this figure we will see the low-level images that we use for ground truthing and how they compare to the orthophoto with 7 centimeter resolution on the right. And this is the same site in 2 consecutive years for Parthenium, where you see the changes in the population size. And it needs to be noted that the timing here is crucial. You need to capture it in the same phenological stage, because like you see on this picture on the right, the massive floods last year here in Kruger actually washed away the invasive populations, but they were coming back after this. The same example for Xanthium. Here we see the map of the same place 2 years, 2022 and 23, and it says 6% over that area, which is 250 by 500 meters. So it doesn't seem much, but if you realize that the river, the actual riverbed is about a half of the picture, even less, and it also includes the water surface, then you get to some 15, maybe 20% cover of the side, which is definitely something that can have impact. Datura, the same thing. Here you see the distribution design in the form of small patches that are sort of moving from year to year. So what is this all good for? Once we have the data processed, we hope that we will be able to identify factors that determine the size and distribution of invasive populations in relation to riverbed morphology, stream features, and also propagule pressure. I've already mentioned the fine-scale interannual population dynamics that we know it happens, but we don't know much about how exactly. And together, all the information from the project we hope will provide a basis for modeling future invasive species dynamics and their impacts in Kruger Park. Thank you for your attention.
Very much. I fear no time for questions. So the last speaker in this session, and to wrap up the theme and just again thinking about trickle-down effects of invasives. So Clara is speaking to us about the influence of invasives on mammals.
Okay, good afternoon. I'm sorry to keep you. You look tired, but I have a lot of pictures in my presentation, and it's about the good kind of herbivores. So, okay, so my name is Klára Pišková, and I'm from the Czech Academy of Sciences. I don't think I need to say anything else because you heard everything already. So I'm going to focus on the herbivore part and on the impact of these invasive species on herbivores here in Kruger. So we all know that rivers are really important for the functioning of the savanna ecosystem, and they are very important also. They play a key role in the survival of animals, in our case herbivores. For some of them it's the habitat that the animals actually live in, or they come to drink, swim daily. Obviously there's food to eat, there's shade to rest in, and then in the riverbeds there's a lot of these patches of green vegetation throughout the year that become very important for the herbivores to also come feed on. But we also know that rivers are an important pathway for the introduction of invasive species. So what happens if we have an invader that comes in and it starts spreading and it becomes dominant, and then eventually it can start displacing some of these native species? And what happens to the animals? Does it maybe make the habitat unsuitable for them or less attractive? So that's what we set out to find out. And okay, I don't think I need to introduce these species anymore. Just to show you what it looks like if they become really like spread in some areas. So this is Datura. I think this is in Xinguezi River. And then Santium, a lot of you probably have seen this. This is on Letaba, and you can get really, really big areas, as you already heard again, covering big parts of the riverbed, like continuous growth. And the same for Parthenium. It doesn't grow in the riverbeds, but on the banks, but it also makes these very, very thick, continuous growth that is very unpleasant to walk through. So we already heard about the effect these invasive plants have on the soils and on the native plant communities. So now let's have a look at the herbivores. So to find out, we asked these two questions. The first one being, how do herbivores utilize the invaded habitats? And do they differentiate between the invaded habitats and the uninvaded? Do they maybe prefer to avoid one or, yeah. And the second question is quite easy, do they eat them? So we use the same setup as for the soils and plant survey, so I don't think I need to introduce much more. And at those sites, we did two types of fieldwork, the first one being camera traps. And we set 42 camera traps. We always had a pair of an invaded site with an uninvaded, like a control site. We set the cameras on a field scan settings, which is basically like a time lapse, so it took a photo every 5 minutes, or a few photos every 5 minutes. I'm going to show you results from the first 2 months of the project, which is from rainy season of last year. And we, as I said, we use this to gather data on how the animals utilize the habitat, meaning not only if they like walk through the invaded areas, but if they or uninvaded areas, but how much time they spend there, if they graze there, rest there, and so on. And another thing that we used this data for was, or the camera traps for, was to monitor the change of cover of the invasive species, meaning that we could see how they grow from little seedlings to big plants and kind of dominate the area. So the first thing that we found out, which was quite interesting to us, is that in many sites we found Datura and Xanthium growing together. And it did seem like Datura would come first and then would slowly get either displaced by Xanthium, or it could be a different phenology of those plants. We have to look into that. But they did seem to kind of grow one after another. So this is the cover that I mentioned, and it's a sum of all the invaded sites together, and so it's like a median cover. And it does kind of reflect what we see in the field. So the first one is Datura, and it has the smallest cover, and it does grow more in like a patchy pattern as opposed to Xanthium and Parthenium, which, as I showed in the photos, that they tend to make these like really huge growths. Now, I say cover, but it's not an actual cover from botanical data. And we, because we use the camera traps to get this cover. And so what we did, we would cut up the photos into a grid. It's not the exact grid, but it was something like this. And first we would remove the areas that are not of interest, where the plants can't grow or the animals can't walk, which is usually the sky or the areas very, very far away. And then with the remaining part of the photo, we would count or mark each square where the invasive species is present. And we would do this, we would take one photo per day for every camera trap and mark all the squares in all the photos. That way we could get this change of cover in time that I showed. And you can see that not the whole area that the camera trap is taking photos of is full of, or is invaded, or is overgrown with the invasive species. So to know how the animals utilize the habitats, as I mentioned, we would count the number of the animals, and then we would say if they are in the invaded area or uninvaded. So here, for example, you'd have 8 waterbuck in an invaded patch. If they were more to the right in the uninvaded, then they would be in an uninvaded patch. I hope it makes sense. So we took this data to get an answer to this question, and this graph shows the utilization, or you could say like habitat preference, for Xanthium. Unfortunately, we don't have data for Parthenium, which seems to be the worst one, but so far I can only show you Xanthium. So the purple or the dark color shows the presence of animals in the non-invaded habitats. And it's kind of like a combination of the number of animals, but also how much time they tend to spend there. So if the animals will spend like an hour in some areas, then it will be reflected in the graph. While we can see that hippos, impalas, kudu, zebra, they don't really make any difference, or they don't differentiate between whether the habitat is invaded or uninvaded, buffalo and elephant, they really tend to avoid Xanthium. And this is basically the same thing, although it's done on a more coarse data, but I wanted to show because it does have waterbuck. The top row is the control site, so we can ignore it for now. But in the bottom right corner, you can see the data from the invaded sites by Xanthium for waterbuck. Now the bars show number of animals per day, and the blue color is if the animal was present in the non-invaded part of the invaded site. And the yellow shows if it was in the invaded patch directly. And you can see barely any yellow with the waterbuck, so they also seem to really avoid Xanthium. This is from Xinguetzi, and it's Xanthium. I counted about 12 lions in there, and there's probably more hiding that we can't see. So maybe that's the reason why waterbuck don't want to really go there. And this was the second part of the fieldwork, to find out about the herbivore diet. And so we put on some gloves and went to collect some dung. And again, metabarcoding. And we wanted to see if there is any of these 3 invasive species in the diet. We focused mainly on these 5 species, and the reason for that is because they are often found in the riverbeds or around the rivers. So if anything is likely to feed on these invasive species, we would think these ones. And also it's much easier to find the dung of these animals and to recognize it. So we collected so far almost 300 samples of dung. It's not really evenly distributed across the park. We're going to collect more starting Friday. And we got over 500 OTUs, and 344 out of those could be identified to species level, belonging to 81 families. These are the families, and I'm not going to spend too much time on this. It's just to show you what we got. The most commonly consumed families were legumes and grasses, which I suppose is not surprising. So, to answer this question, now this is an elephant that has a mouth full of xanthium. So we got really excited when we saw this. We thought, okay, now the elephants will rescue Kruger from the invasion of xanthium by eating all of it. And let's see. So this is the number of the samples that had any amount of the Datura Parthenium and xanthium in it. So nothing eats Datura. Almost nothing eats Parthenium. There is 6 impalas, but I think they probably just tasted it by accident. And we didn't have many impala samples anyway, so. Xanthium is a little bit more interesting. So almost half of all the hippo samples had xanthium in it, about 40% of impalas, 13% of buffalo, and about a quarter of all elephant samples had xanthium in it. So that looks more promising. But if you look at the actual amount of the plants in the samples, it's nothing. So unfortunately, elephants or any other species will not save the park from any invasion. So to answer the questions, no, the herbivores do not eat the invasive species. So to sum up, some herbivores, such as elephant, buffalo, or the waterbuck, they seem to not like the areas that are invaded by Xanthium strumarium, while other species don't really seem to care, like hippo and impala, zebra. As I just said, the herbivores don't really eat the invasive plants, and if so, it's just probably by accident, or they are curious about what's growing there. And if they do, it's in a very small amount. There was one hippo that had over 60% of xanthium, but that's, I don't know, it's probably hired by park management to do control. But there was probably 10 species that had more than 5% of any of the invasive or xanthium in it, so it's basically nothing. These 3 invasive species that we studied definitely do not serve as a food source for the herbivores, or at least the ones we sampled. And we do know that the plants do have some impact on the native plant communities. So in that way, it could somehow indirectly affect the herbivores, because their natural native food sources might change a little bit or be less abundant, while the new species do not replace it or serve as a new source. And for those reasons, species that are more closely dependent on the river habitats are likely to be more impacted by the invasions. And that's it for me, and I would like to thank you for staying awake, and game guards and everyone who helped. Thank you.
Thank you very much, Clara. Any questions for Clara at this point? End of a long day. So just a big hand of applause for all the speakers. Just thank you very much. So just I guess one little bit of housekeeping. I think, Sam, correct me if I'm wrong, the buses are leaving at 6:20 and dinner to be served at the golf club. So thank you very much for a very invigorating session and see you guys at the cricket club. What did I say?